Hello dear students, welcome to another interesting lesson from Dr. R's Biology. If you are new to this particular channel, you can click on the subscribe button and bell icon so that you will get updates on new uploads of videos. In this session, we are going to discuss some interesting fact with regard to the genetic codon. So you can see the question there. Initiation codons for protein synthesis are. So, and you are finding more than one option. Whatever we have learned so far is actually AUG. Most of you are aware of only one codon that will act as the initiation codon, that AUG. Now in the given option you can find at least two in every option. So which one you will choose? So another question, the GUG specifies amino acid valine. However, when functioning initiation codon, it specifies. So you are getting an information from this question that GUG is also acting as an initiation codon. So which that will be coding for when it is acting as the initiation codon. Let us see the answers later. Now let us move to the next question. Sometimes the starting codon is GUG in place of AUG. So GUG normally stands for. So it's a direct question from the genetic codon table. So let us see the answers later. And then another question. The most common start codon has dash in the corresponding DNA sequence and dash in the template DNA sequence. So you are getting an indirect question directly from the table that is given in your textbook, NCERT textbook, with regard to the genetic codon. So finding answers for all these questions, you can just read a research article that was published in the research journal Critical Reviews in Biochemical and Molecular Biology, and that was published in 2012. And the title of the article was Dual Functions of Codon. And I will be discussing the major points that is discussed in this research article with regard to the codons, genetic codons, and related to the NCERT syllabus as for the NEET examination. Okay, I will not be reading the entire paper. Instead, you can, if you are interested to read it completely, you can just go to the NCBI website, write the title or type the title of this particular research article, and you can get the free full article. You can read that. Okay, so let us see the main important point that is being discussed in this particular research article with regard to the genetic code and for the NEET examination. Okay, so I highlighted this particular thing here. You can find. Okay, so you can see that. So duality of codon function is known in the use of AUG. In translation, that you know that methionine. Okay, right? Because methionine is acting as methionine coding. AUG is acting as the one which codes for methionine as well as the one which codes for initiation of this particular translation process. Okay, so and now in case of this selenocysteine and pyrrolysine insertion codon, okay, you can find that this particular two, selenocysteine, that is not new to us, selenocysteine and pyrrolysine, that can be coded by codons UGA and UAG. So you are familiar with the UGA and UAG because it is acting as the stop codon in normal cases, right? Because UGA, we know it is known as the opal, and the UAG, that is known as the amber, right? Amber and opal. So you are familiar these two as the stop codon that will be terminating the translation process. Instead, you can find its dual function as it is coding for selenocysteine and pyrrolysine. UGA is that opal is coding for selenocysteine, and the UAG, the amber, is coding for pyrrolysine. Just keep this point in mind. Selenocysteine and pyrrolysine can be coded by stop codons UGA and the UAG. Keep in mind. Now, in the same article, you can find another interesting part. So, methionine dual function plus additional initiation codon. So, here we can find AUG act as an initiator of protein synthesis as well as a codon for methionine. Incorporation at internal protein positions in eukaryotes and N-formylmethionine in prokaryotes. So you can find that AUG will be coding for methionine. methionine normally in case of the eukaryotes and archaebacteria, and it will be coding for formylmethionine. Okay, formylmethionine in case of the bacteria. Okay, in case of the bacteria, and then in case of the mitochondria, and in case of the plastids. So it is adding formylmethionine as first amino acid. Okay, first amino acid, not else. And within the genetic sequence or the protein sequence, or if it is internal position, the AUG is standing in internal position, then it will be coding for methionine itself. Okay, it will be coding for methionine itself in all organism, including mitochondria, plastids, bacteria, eubacteria, archaebacteria, and the eukaryotic organism. Keep that point also in mind. Now, additionally, in case of eukaryote, okay, in case of the eukaryote, we can find here, okay, we can find here. So in prokaryotes, the use of GUG and UUG as initiators in addition to AUG. So you are finding an extra 2. In question you have found only GUG, and then in addition to GUG another start codon UUG that also act as the initiation codon in case of the prokaryote in addition to AUG. So if AUG is absent, then they will be reading the GUG or UUG as the initiation codon, and at the same time you have to keep in mind that there will be coding for methionine itself. Methionine itself, or whatever the first amino acid. For example, in case of the prokaryotic organisms, you will find a formylmethionine as the first amino acid in a protein. So in case of that, formylmethionine will be added. So whatever the initiation codon is being used, in addition to AUG, the coded amino acid will be definitely this methionine. Okay, so it will be definitely this methionine. So don't forget that particular thing. Now, if you're taking an example in case of the E. coli bacteria, you can find that almost 40%. So in addition to AUG, AUG will be coding more than 80% initiation codon portions. 40% of the initiation part in most of the genes will be GUG, that is acting as the initiation codon, and in 3% you will find a UUG as the initiation codon. So it was an article published in 1997 that was done by the Blatton et al. Okay, Blattner et al. And you can find that article too either in the Google Scholar. So if you're interested, you can just read the full article, but in the NEET point of view, I have shared the entire content that you have to keep in mind before going for the NEET examination, and in the biology part, you can appear this question. Now, I'm concluding all these things with one more slide. So whatever we have told so far, I'm just concluding in some points. So what are the points that we have discussed? The start codon always codes for methionine in eukaryotes. So it will be always coding for methionine, whatever it may be. It can be AUG, it can be GUG, or it can be anything. Okay, so it will always codes for methionine in eukaryotes, archaea, and it will be always coding for N-formylmethionine in case of the bacteria, mitochondria, and plastids. Keep that point in mind. Now, alternate start codons are still translated as methionine when they are at the start of the protein, because a separate tRNA is used for initiation. So why methionine is always added regardless of what type of initiation codon is used? The answer is a separate tRNA is always used for initiation of translation process. That's why the reason AUG is coding for methionine, GUG is coding for methionine, UUG is coding for methionine. Everything that acts as the initiation codon will be coding for methionine in case of the eukaryotes and archaea bacteria, and formylmethionine in case of the bacteria, mitochondria, and plastids. Clear? Now, alternate start codons. Okay, non-AUG codons are very rare in case of the eukaryotes. The GUG, UUG, etc. we found are especially in case of the prokaryotic organisms. Okay, so it is very rare in eukaryote, but still research is going on to find out the eukaryotic non-AUG initiation codons. Let us see later. Okay, so we are just focusing on prokaryotes. The NCERT book also is focusing on prokaryote with regard to the transcription and translation, and they are just mentioning some additional points with regard to the this transcription or central dogma with regard to eukaryotes. So we are just focusing on the prokaryote itself. So in case of the prokaryotes, the alternate start codon significantly or mainly they are coding GUG and UUG. Keep those point also in mind, the GUG and UUG. So GUG is main. So suppose if you are finding only single solution, single option, and there appear one option as GUG and second option as UUG, you have to choose GUG first because that is the main initiation codon after AUG. Clear? So the preference should be given to GUG first and then UUG to second. Now, as I told in your call, more than 80%, 83% is AUG initiation codon, 40% of the genes will be coded by GUG initiation codon, and 3% of the genes will be coded by UUG initiation codon. Okay, so the mitochondrial genomes. So we had to focus on that too. Some additional alternate start codons are more significantly used. What are they? That are AUA and AUU in case of the human mitochondria. Keep in mind, AUA and AUU are some additional non-AUG initiation codons that is present in the human mitochondria. So these are the main points that we have discussed in this particular lesson so far. So now let us go back to the questions and we can find answers for the same. So the first question. What is the first question? Initiation codons for protein synthesis are. Now you know the answer. What is that? GUG and AUG. In addition to that, what you have to find? It's actually UUG2. Clear? Now, GUG specifies amino acid valine. We know that because you can remember guava. Okay, because the codons that is starting with the GU will always code for valine. So GUG or whatever it may be, it will be coding for valine. Normal case, that is a valine. But when the GUG comes as an initiation codon, it will be specifying. You know that whatever the condition is, it will be specifying methionine. And suppose in the same question, the option appeared is methionine, and in case of instead of isoleucine, you found formylmethionine. What you will find. So you have to choose option D here. Why? Because GUG is actually the non-AUG initiation codon that is present in case of the prokaryotic organism. Okay, prokaryotic organism. So as a result, in case of the prokaryote, the first amino acid due to initiation codon will always formylmethionine. So you have to choose formylmethionine if it is available in the option. Or otherwise, what you have to choose, you can just choose the methionine. So whatever the start codon is, you have to choose methionine or formylmethionine as the amino acid in the first position of a newly formed polypeptide. Now, next question. Sometimes the starting codon is GUG in place of AUG, GUG normally stands for. Okay. Sometimes the starting codon is GUG in place of AUG. Okay. GUG normally stands for. GUG, we know that the GUG normally stands for valine. Okay. Right now, we know that it's because of the guava that you are familiar. Okay. Valine is the one which is coded by GUG codon in the normal condition. Only when it is at the initial position, it will be coding for methionine or formylmethionine more specifically. Now, the most common start codon has dash in the corresponding DNA sequence and dash in the template DNA sequence. Which is the most common start codon? We know that the most common start codon is actually AUG because that is coded or that is present in 83% of the E. coli genome in an experiment. So it is the most common initiation codon. So it is AUG. Then the most common start codon has dash in the corresponding DNA sequence. In the corresponding DNA sequence means what? What is actually mean by corresponding DNA sequence? It is actually mean by the coding strand. Okay, coding strand. We know that coding strand is not coding for anything and it will have exact similar sequence with same as that of the mRNA. So the coding strand will have a what instead of AUG. We know that if it is A, it will be definitely thymine, right? If it is U, it will be definitely adenine. If it is G, it will be definitely C. So TAC will be there in the corresponding DNA sequence. And what is the second part of the question? Dash in the template DNA sequence. Okay. The template DNA sequence will have this particular thing. And in a coding sequence, it will be exactly same. Only what? A, instead of U, what will be there? Thymine will be there. And instead of G, what will be there? G will be there. Because we know that the coding strand will have exact same mRNA except uracil thymine, that combination. Okay, so ATG will be there in the corresponding DNA sequence, and in the template sequence, the complementary one, TAC will be there. So what is the first option is the answer, ATG and TAC. You can easily find out. And the similar kind of question can appear in a different way. What? The second most common start codon has. The second most common. Some additional point is added here. Second most codon start codon has dash in the corresponding DNA. What is the second most common? The second most common initiation codon is nothing but GUG, because it is appeared in the 40%, right? So GUG. So GUG will have a corresponding DNA sequence. What? G, T, G. GTG will be the corresponding DNA sequence. Okay, and what will be there in the template strand corresponding? C, A, C. Okay, so what will be the option if that is the question? Option will be C if that is the question. Clear? So the same kind of question can appear with the third common too. Okay, so it can be the 3% of the genes in E. coli is initiated by the expression of into polypeptide is initiated by dash codon, and it will have dash in the corresponding DNA sequence and dash in the template DNA sequence. With the 3 parts, you can have the same question. Okay, so be careful about these kinds of question and be aware of the fact that AUG 83%, GUG 14%, and UUG 3%. Clear? So that's about the session, and thank you for listening to Dr. R's biology. Okay, thank you for your time, and let us see later with some other interesting lesson. See you, bye.